I think also this Pratik, you were asking about that internship thing, right? The previous one. Okay, so actually, they contacted me. They asked me to even directly write a mail to them or the concerned authorities because I don't think I will be the one to answer this question. So they asked you to mail them directly or contact them, and it will be answered back to them. So we'll start the class. Yes, yes. This is Becky. Is this okay now? Is there any background noise coming or this is fine? Okay, I think there's a little disrupt in the connectivity probably. Um, if it's too bad, just let me know in between. Okay. We'll start. The screen is visible, right, to all of you? Oh, okay. So, this tutorial session, this is the we will summarize week five's lectures and the content, and then we'll look at few example questions. So here, like last week, we were studying about how to clone a certain genetic gene sequence. Okay. Now, after cloning the gene sequence, we were introducing it into uh, we were telling like, okay, we are making recombinant vectors, and then we have to introduce into an organism. Suppose it can be plant, it can be bacteria, it can be yeast. Okay. So we were introducing the recombinant vectors. So now in that introduction, there can be various types, various mechanisms of that particular delivery of that recombinant vector into the host. Okay. So what exactly happens here is so this is the donor cell. Okay. And in general, when we are talking about the delivery of a certain recombinant vector from the donor cell into the recipient cell, then the, the first and foremost thing that should be kept in mind is the surface chemistry of the host cell and the recipient cells should be compatible. Now, host cell surface, especially it depends on the charges. Why? Because especially the structure of the cell membrane, the constituent or composition of the cell membrane, the phospholipid and the protein content and all those components of the cell membrane can differ from one organism to another. Okay. So that surface chemistry should be compatible or specifically by compatibility we mean here regarding the charges. Okay. So if DNA due to its phosphate, due to the phosphate groups or phosphate backbone present, it is negatively charged. So the like the property or the character of the cell membrane that is there, that should be amenable for like it should not repel the negatively charged DNA too much because otherwise then introducing that particular DNA into the host cell would be difficult. Or what one can do is you can always if suppose we need that particular recipient cells, even though the suppose uh, the charge distribution of the recipient cell is negatively charged, right? So what we have is uh, Okay. So, is this better now? Okay. So, from where should I repeat, or where did it go bad? Connectivity? No, ma'am, you can open here. Okay. So, what I was talking about, now suppose, now presence of cell wall in case of bacterial cell, fungal cell, or a plant cell can cause or can provide an additional physical barrier that can interrupt with the uptake or the delivery of the genetic sequence that we are talking about. Okay. Because charges on, see, this is what is written here, like negative charge, which is given by the phosphate backbone on DNA, modulates the interaction with the host cell, especially on cell surface. So that's why when we want to introduce a foreign DNA or a recombinant DNA, then we have to make the recipient cells competent or compatible enough so that it can receive the DNA. So the recipient cells in this case are called competent cells. We have to make the cells competent. Okay. Now, now we will go a little bit like uh, look at the DNA delivery and making cells competent, as well as how to how to really deliver the DNA or recombinant DNA into the host cell uh, in a little bit more detail. Okay. Now, usually what happens is divalent cations, especially calcium, magnesium, manganese are also used, but specifically divalent cations, calcium, Ca2 plus, has been the most effective chemical treatment to bring about transformation. Now, among the different cations that have been used, divalent calcium cations have been proven to be the most effective one. So all of this, what I am telling is, all these have been experimentally done by in research laboratories, and these have been experimentally proven. So people have tried out, different, not only cations, I mean divalent cations. People have tried out monovalent ions. People have tried out trivalent ions also to, see if uh, like which kind of treatment, which kind of chemical treatment made the cells more competent. Okay. So, or they have also used like combinations of not only divalent cations, different types of divalent cations. They have also used combinations of trivalent cations, divalent cations, monovalent cations, or they have also used some carrier molecules to carry the DNA into the, like, to the host cell. Okay. Now we will look at all of these one by one. All right. So first, let's look at divalent cations. Now, combination of divalent and monovalent ions like calcium and manganese, calcium, rubidium, then there's DMSO or dimethyl sulfoxide, and there are various other alkaline metals where where we uh, incubate after treating the cells. Nothing, like if you put the cell suspension in a solution of these in a solution of salts containing salts of these ions, what I'm talking about, calcium, magnesium, manganese, rubidium. Okay. And when you incubate this at 0 degree, or actually we are talking about ice here. Okay. So these different combinations have proven to be effective, or have proven to be effective in making the cells competent. All right. But most of all, the most effective one was found to be the divalent ions like calcium, and then magnesium and manganese have also been proven to be effective. So generally, in general, like all divalent cations enhance the transformation process. Transformation process means introduction of genetic material of foreign DNA into the host cell. Okay. Now, presence of magnesium in bacterial culture media increases the transformation efficiency by up to 15 to 20 times as compared to the cells grown in the absence of magnesium. All right. Now, what is this concept of transformation? It's basically This is the donor cell. Okay. And the particular DNA from the donor cell is isolated, and then that particular isolated genetic material is uh, delivered or introduced into the acceptor or recipient cells. Now here, suppose this donor cell, what I was talking about, suppose this donor cell is made resistant to certain antibiotic, ampicillin resistant gene has been introduced here. Now, if that particular gene, ampicillin containing the ampicillin resistant construct, is isolated and then introduced into a recipient cells which ideally isn't ampicillin resistant or resistant to ampicillin, then if that gene is getting delivered correctly into the recipient cell, then what will happen is the recipient or acceptor cell will also gain resistance to that against that particular antibiotic. Okay. Now, this is if we are talking about transformation, then we should definitely be talking about Griffith's experiment. Okay. So what was done, like like how did this idea come up that okay, something is getting transferred from one organism or from one cell to other cell? Okay. So what happened was, Griffith was there in the 18th century. What happened was, uh, the, like Streptococcus pneumoniae, this bacteria has two strains. One is the virulent strain and one is the non-virulent strain. Now, virulent strain has a smooth outer layer in the bacteria, and the rough strain, and that particular due to the smooth outer layer, this is called the smooth strain, and this is the virulent one, which can actually kill and kill the kill the organism. And in case of non-virulent strain, which is the rough strain, when this particular rough strain of the bacteria were introduced into the mice, mouse, the mouse can live happily. Okay, but then when the virulent strain was introduced, it died. Now what they tried that okay, let's kill this virulent strain. So they heat killed the cultured cells and then injected that particular sample inside the mouse and found that the mouse was still living. But when they mixed the rough strain, which was actually non-virulent, and the heat-killed S strain, okay, smooth strain is also known as S strain and rough strain as R strain. Okay, when this rough strain and the heat-killed S strain was mixed together, and that particular sample, this mixture was introduced into the mice, then it was found that the mouse was dying. But when only rough strain or R strain was being introduced, mouse was living happily. Only when the heat killed S strain, because uh, without, like, as in when the only heat killed S strain was there, mouse was living happily. But when both of these were mixed, the DNA from the actually now we know that the DNA from the this particular S strain could really get inside this R strain cells and made it so introduced that virulence factor inside the R strain cells that when injected inside the mouse killed the mouse eventually. Okay. So this at that time DNA wasn't known as the genetic material or the like the thing that is getting transferred from one cell to another cell. So Griffith coined the term as transforming principle. Okay. In this case, as something is getting transformed from the heat killed S strain into the R strain cells. All right. Now let's talk about the mechanism of transformation. So transformation is basically the process by which a cell-free DNA, which has been isolated, suppose from the donor cell, is taken up by another cell. Not necessarily bacteria. It can be some other organism also, some other yeast cell. It can be plant cells also. All right. So the DNA from donor bacteria binds to the competent recipient cell and DNA enters into the cell. Okay. The DNA enters into the recipient cell through an uncharacterized mechanism. This particular uncharacterized mechanism, like one we discussed about usage of divalent cations. So we will look at the different steps that are being used during transformation. And till now, till date, like exact mechanism, like there's a protocol for introducing or for delivering this genetic material or recombinant DNA into host cell, which is transformation. There is this transformation protocol, but till now, like there are various different methods. Like basically, it's the treatment with divalent ions as well as treatment with divalent cations, basically, and incubation at like different temperature changes. Okay, like exposing the cells to the to different kinds of temperature changes. So using these two methods, what happens is the pores are created in the cells, recipient cells, and then the DNA can be taken in easily. Okay, but it's still like there are different kinds of explanations as to why this particular process is carried out. So we will be looking at each explanation, like different kinds of explanation of each protocol as in each step of the protocol for transformation. Okay, in the upcoming slides. Now the DNA is integrated into the chromosomal DNA through homologous recombination, and naturally transformation is common between closely related species only. Okay, because the compatibility needs to be taken care of between the donor as well as the recipient cells. Okay, in case of natural scenario, in artificially, suppose in laboratory work or in general, what we do is like we can artificially make the recipient cells compatible with the donor cells so that it can accept the donor cells DNA or in general genetic material. Okay, so what is happening here is this is the donor cell. We have extracted donor DNA, fragmented it after cell lysis by chemical or mechanical means. So we looked at the different ways to lyse the cells, all right, mechanical or chemical lysis. And then the donor DNA fragments bind to competent recipient cells. Okay, one strand of donor DNA degraded on binding, and this is the competent recipient cells. And this eventually is taken up by the recipient cell and gets integrated into the genome of the recipient cell. And then when the cell division or when the recipient cell divides, then of course DNA will be replicated, and eventually this donor cell or donor DNA gets incorporated into the genome. Okay, so some of it might take it up, some of it, suppose even if it's getting if it is getting integrated, then it might not be replicated or transcribed, okay, selectively by the, suppose in bacteria, what many times happens is in E. coli especially, what happens is sometimes when you introduce a foreign DNA, unless suppose we want to express a particular eukaryotic gene in E. coli cell, sometimes what happens is even when the E. coli has taken up the genetic material, like like genetic material foreign gene encoding the eukaryotic protein, suppose. Okay, it might not transcribe that particular sequence because uh, the cell doesn't need it, or it might cell doesn't need it, and also like it might add to the burden. Okay, or it might, because that is a eukaryotic protein. What will happen is the protein, when transcribed or expressed, like transcribed, translated, then what might happen is it might prove toxic to the bacterial cell also. So bacteria can choose not to express that particular genetic sequence. So again, there are many different kinds of modifications that can be done, or again, the recipient cells can have, the E. coli cells in this scenario, what I'm talking about, can have additional, sequences attached, additional uh, genes or sequences attached, which can actually help it to code for certain factors which will help in uh, transcribing as well as translating the eukaryotic genetic sequence. Okay. So in this way, we can like really play around with the recipient cells genome, as in recipient cells genetic composition, and which can actually our uh, help in carrying out our experiments or our laboratory purposes. Now, what happens is the repulsion between the foreign DNA and the bacterial cell. Why? Because owing to the negative charges on them both are overcome by divalent cations, calcium, magnesium, manganese. Okay. This is applicable for linear DNA fragments as well as circular DNA molecules, all right, like plasmids that we were talking about. Now, divalent cations can actually bind both to the cell because of the surface charge properties as well as the DNA, negatively charged DNA, thereby neutralizing the charge altogether. Okay. Now, what happens is the calcium bound to the DNA can further help the DNA to get absorbed onto the competent cells. All right. And also, DNA binding proteins, which are present in the cell membrane, can also help in this particular interaction. Okay, if there are DNA binding proteins present on the cell membrane, then that can actually help the DNA to really dock onto the cell surface or recipient cell surface. Okay. Now, this DNA binding to the membrane can eliminate the risk of detachment or expulsion of DNA definitely if like only if there are no proteins present to really receive or interact with the DNA, okay, or any DNA binding proteins, then this will be what to say, a transient attachment of that DNA. So it can really just due to the covering or due to the interaction of divalent cations with both DNA as well as the cell surface or the cell membrane, what might happen is that particular interaction will be transient, okay, which can detach the DNA and then the uptake will be poor. Uptake of this foreign DNA by the recipient cells will be poor. Okay. Now, what happens is we lower the temperatures, okay. So at low temperatures, basically like ideally we use ice. Okay, we always do everything in ice. We put the cells in ice, add DNA in like in an ice bucket, everything. Okay. So the low temperatures used in transformation protocols congeals the lipid moiety and consequently restricts the fluidity of the cell membrane. The fluidity of the cell membrane is basically provided or basically comes due to the lipid lipid molecules present in the cell membrane, right? So this actually, if the fluidity of the cell membrane is limited or restricted, then this will strengthen the calcium cell surface interaction definitely. If calcium is binding or interacting with the cell surface moieties, cell surface moieties present in the recipient cell, then if that particular moiety is moving, suppose it is so the calcium, these cells are actually resuspended in a solution containing calcium chloride, magnesium chloride. Okay. So what happens here is now this is these calcium ions are actually getting deposited on the cell, right? So not exactly precipitated, like in general, that will interact with the cell surface. Okay. Now, if the cell surface moieties are only or cell membrane moieties are moving, then this will again, lower the strength of the interactions between calcium ions or the divalent cations that we are talking about and the cell surface, right? So so at lower temperatures, what happens when we do the things on ice, then that will actually restrict the movement of these lipid molecules present in the cell membrane. Okay. So in this way, calcium ions bound with the cell surface as well as the foreign DNA can bring the DNA to the cell. Okay. Now, relative association of divalent cations has been found. This again has been experimentally proven that relative association of divalent cations is more with the cell membrane as compared to its association with foreign DNA. Okay. Now, what happens is membranes can absorb calcium very readily, and once the calcium gets inside the cells, then it can be neutralized by the membrane phosphates. We were talking about phospholipids also, right? So these can get neutralized by membrane phosphates present on the cytosolic side. Okay, definitely, like this is a lipid bilayer. So there's like towards the like cytosolic side, there are things, and between there's this cytoplasmic side or cytosolic side, and there is the out like extracellular region or extracellular environment. Okay, that the lipid moieties interact with. So the binding of calcium ions to the membrane can also cause changes or can also lead to changes in membrane permeability. Okay. Now, what happens is whether you are treating the cells with divalent ions or trivalent ions on ice, uh, there's a protocol. In the protocol, we give a heat shock to the cells. Okay. So at like the it is at 0 to 4 degrees Celsius. The other steps are carried out. and then suddenly for around 90 seconds, what we give heat shock at some raised temperatures, ideally 42 degrees Celsius. Okay. So this actually creates a temperature imbalance. Hmm. Now what will happen is molecules which are having increased motion, Brownian motion in this case, which are outside the cells, so they're constantly in motion. So what those particular molecules will do, they will help in pushing the DNA inside the cells. All right, because that sudden increase in temperature will disturb the it- not only disturb, like increase, lead to increased motion of all the molecules around, and then that will help in pushing the DNA inside the cells. But then again, it is still unclear if this particular force that we are talking about is sufficient enough to push the DNA molecules are already outside the cells, right? And then with increased temperature, what people have speculated that the molecular motion around, increased molecular motion around will push the DNA molecules inside the cells. But then, is it really enough that pushing force that is created by these molecules onto the DNA molecules, if this is enough to push the adsorbed DNA molecules inside? Now, this is the group that was studying all these, especially with respect to the temperature changes, how different kinds of temperature changes can affect the competency of a recipient cell. So they actually studied the efficiency of cooling and heating cycles by increasing the number of cycles until maximum transformation efficiency was achieved. Now, this term transformation efficiency is coming up multiple times. So we will again look at after like looking at all the principles and procedures, how exactly this transformation efficiency is. What do we exactly mean by transformation efficiency and how this is calculated? Okay. Now, when we lower the temperature, that can actually contribute to protein loss. And when we are heating the uh, cells, like we were talking about heat shock, that can actually lead to lipid loss. So together, this lowering of temperature, increasing of temperature, this temperature variability is increasing the transformation efficiency because the pore sizes are enlarged eventually. Okay. Also, due to loss of lipids and proteins, membrane gets depolarized, thereby reducing the repulsion between the DNA molecule and the membrane. Okay. And cell density can also affect the efficiency of transformation. Okay. If this there should be a proper ratio between between how much of foreign DNA has been introduced and how many cells or what is the concentration or the number of cells in a particular volume present to take up that foreign DNA. So the cell density can also affect the efficiency of transformation, and it has been reported that maximum competency is observed at cell density ranging from around 107 to it should be, I think, 10 to the power 7 to 10 to the power 8 cells per ml in the log phase. Okay. It should be like 10 to the power 7 and 10 to the power 8. Okay. It can't, this is too less if we are talking in the range of hundreds. Now, but the question still remains whether the pores that we are talking about, through which the foreign DNA will be entering eventually, are formed by the calcium treatment or whether they are actually naturally present. Okay. So again, some scientists uh, looked into this and they actually saw that natural channels exist already, which are called Bayer's bridges. Just no need to look at the term. This is just the bridge or natural channels for um uptake of any foreign materials or passage of molecules between the cells to the outside environment or uptake of molecules from the outside environment, extracellular environment into the cells. Okay. So there exist natural channels, often called Bayer's bridges in the membrane, that can serve as potential pathway for DNA. Okay. Now, just imagine it goes way back to 1980s, which is Hanahan stated that the competent cells can have many sites or channels, and all these sites and channels have an independent chance of taking part in the uptake of DNA moving towards the process of transformation. Okay, so see, calcium treatment, temperature variations, are creating the pores, but those might not be sufficient enough for the cells to really take up the foreign DNA. So cells can already, competent cells or the recipient cells that we are talking about can already have naturally present like sites or channels or bridges through which DNA can be taken up. And all the cells, whether these are competent or not, compete for the uptake of plasmid DNA. But if only competent cells are used for the transformation, definitely. If in a mixture you have both competent cells and general, like the general recipient cells, then the uptake of plasmid, they will be competing for the uptake of plasmid, and the efficiency of transformation will be less in that scenario. But if we are only giving culture a solution of competent cells, then definitely we can expect a large number of cells to have the large number of cells to get transformed and have the foreign DNA taken inside. Okay. Now the DNA uptake factor is sum of all the probabilities of DNA uptake through each channel. Okay. Now it has been reported that the chances of transformation are not really increased by increasing the DNA concentration, but actually if we increase the number of bridges through which the DNA can be taken in, then the transformation efficiency can actually increase. Okay. Moreover, calcium has a dual role in this process also. Definitely, like initially what we saw, it was neutralizing the charge, but it also weakens the cell membrane to produce invaginations. Okay, so that the DNA, the entrance or the uptake of the DNA becomes smoother. Okay, now while it was known that the divalent cations can help neutralize the charge, the complex ions can also serve to produce the static force of attraction within the DNA molecule. So this what will happen now is due to this force of attraction, the DNA molecule can form a compact ball-like structure. Now, if something is compact, we looked at different types of plasmid DNA, right? Like it was supercoiled, it was nicked, or it can be linear. Okay. So what will happen is if it is a compact ball-like structure, specifically if we are talking about supercoiled plasmid DNA, then the plasmid DNA in its supercoiled form will be the easiest to transition through the pores or channels in the recipient cells, and the transformation efficiency will be more in case of a compact, like really compact DNA molecule. Okay. A supercoiled ball-like structure of the plasmid will have more chances of entering the competent cell or recipient cell we are talking about than the extended open circular form. Okay. But if the size of the DNA approaches the size of the pore, then the probability of the transformation will decrease sharply. Okay. Now there are some chemicals like spermidine or other trivalent ions. The size of the ball-like structure of the DNA, when the DNA has to be actually supplemented with a carrier DNA, suppose, or some carrier molecule or carrier moiety, then what happens is the overall size of the the structure that needs to be delivered inside the cells, that increases. Then definitely there will be hindrance at the channel, or there will be barrier, like easy transition will be difficult, and the probability of the transformation of the foreign genetic material that we are trying to introduce, that will decrease sharply. Okay. So again, this if again if we try to optimize this heating and cooling cycles, what might happen is uh, the pores can be enlarged, and if the pores are enlarged, then uh, that particular bigger, like slightly high, like larger structural moiety can also be introduced. Slightly larger in the sense that we are talking about, suppose as compared to a supercoiled plasmid, if some other DNA needs a carrier, foreign genetic material needs a carrier to get entry inside the recipient cell. Now, whether using divalents or trivalents, their concentrations need to be optimized, definitely, such that all the phosphates of the DNA are not rendered inaccessible. Hmm. Now, because some parts of the DNA have to adsorb onto the cell surface, and for that, free phosphates are required. Okay. Now, what exactly, there are some other factors that can really affect your transformation efficiency, which is the transformation efficiency is greatly affected by the type of the host cell we looked at it, because they can have different cell surface structures. And with this surface structure, there can be different kinds of saccharides present, or polysaccharides that protrude out of the cell surface, right? Now, these surface structures can interact with the cations that we are talking about, especially the divalent cations and the DNA, thereby making the cell competent for transformation. Now, there are different strains of E. coli, okay, which have been reported to show variance in transformation efficiency. Why? Because of the differences in the chemical properties of the cellular envelope, okay, present on the cell surface or extending from the cell surface, present in the integral part of the cell membrane, like that. Okay. Because something which is present in the, like suppose integral membrane proteins we are talking about. Now the particular property of that particular membrane protein which is integrated inside the cell membrane, that will also affect how the foreign DNA, that will also affect whether the passage of the foreign DNA that needs to be introduced is smooth or a little bit difficult, or has to be aided using some other chemical treatment or physical treatments. Now, a very dense polysaccharide will actually obstruct the DNA to pass through smoothly. Definitely, but it has also been seen that if we really remove a lot of lipopolysaccharide from the cell membrane using ethanol, then it can also reduce the transformation efficiency, because DNA first attaches to, we were talking about the, like, DNA needs to be absorbed onto the cell surface, right? So the DNA first attaches to some external component of the cell membrane, which can then actually assist, which actually helps in the DNA to really come inside the cell. But if we really try to move a lot of the lipopolysaccharide that we are talking about here, then that actually was seen to reduce the transformation efficiency. Okay. Now, along with density, what is the composition of the polysaccharide we are talking about can also have a role, or can also affect the intake of the foreign DNA. Okay. And calcium ions also interact with the membrane. We talked about it. And this is the concentration that was seen, like 100 millimolar. Almost all calcium gets absorbed by the cell membranes. There is these two moieties present in the cell membrane, right? Phosphatidylcholine and phosphatidylserine. Therefore, the membrane properties can play a major role, like really an important role in taking off the foreign DNA. Okay. Now see. Now, so this is what we were discussing, like physical and chemical treatments, which are used during transformation, like temperature imbalances. We are doing sometimes things on ice and then suddenly giving a heat shock, or raising the temperature, as well as giving, we are treating the cells with different kinds of salts having divalent cations, can help deal with the barrier to pick up any foreign DNA like charge repulsions and pore sizes. Okay. So see here, this is the schematic or the diagram. Like this is the foreign DNA we are talking about, and here the recipient cell is a bacteria. Now, this has been zoomed in, the bacterial cell membrane. Now with this is the bacterial cell membrane, which is negatively charged due to the phospholipid bilayer and other proteins present here. Now what happens is, and DNA is also negatively charged. Okay. Now, when calcium ions are given, this coats the negatively charged DNA, as well as coats the this membrane as well. Okay. And when we really, there are, when temperature variations are done, like sometimes heat shock, sometimes cooling on ice, then what again happens is this fluidity of the membrane is restricted. There can be lipid loss or protein loss, and then so see, 0 to 42 degrees Celsius, there can be lipid loss, and when we are giving a heat shock, then there can be protein loss. So what will happen is the proteins and lipids are basically making up the cell membrane. So what will happen here is this will enlarge the pores or create pores so that the foreign DNA can be taken up easily. Okay. So Due to the calcium ion treatment and temperature treatment, the DNA gets transferred inside the bacterial cell. Okay. Now, how do we really prepare a competent cell? So what happens is bacteria is incubated with divalent cations, like we have been talking about it. Calcium chloride, manganese chloride, or rubidium chloride. Magnesium chloride can also be there for a certain time at low temperature. And what happens during this treatment, during this salt treatment, is the cell wall of treated bacteria swell up and gathers factor which are required for the uptake of DNA, which has been docked or which has been absorbed onto the cell membrane. Okay, and so after competent cells are actually very fragile and has to be handled with care so that it doesn't like in any kind of treatment when you are actually even picking up the cells, the pipette tips and everything should be very gently, you should be very gently handling these cells. Okay. The storage should be proper at like very usually these are stored at minus 80 degrees Celsius in laboratories and the speed of centrifugation, pipetting. So what happens is when you are uh, treating these cells in that solution of divalent cations, what you have to do is you have to be very gentle with your pipetting. You shouldn't be like vigorously mixing the cells. That will actually because these are really fragile. And then also when you are pipetting, you should be gentle. Or what also happens is people, you know, tips, like pipette tips are like this, like very pointed. Okay. So if you really cut it out, then the inlet becomes broader. So the cells won't really, because these are fragile, so the cells would enter very easily through the uh, pipette tips. And then that will, there will be less chance of physical, uh, like any physical harm or physical damage to the cells. Now, this is the general protocol. What is happening here is you have your bacterial cells on an agar plate on a nutrient medium. Then you pick it up and uh, like you transfer it into you culture those cells and then transfer it to a cold. Everything is done in ice and then transfer it to calcium chloride solution and then uh, these cells now in this case sometimes uh, the, the cells that we are trying to prepare can have resistance to antibiotic or it might not have resistance to antibiotics at all. Okay. Now suppose now this is the transformation protocol that we are talking about. So suppose in general the recipient cells aren't ampicillin resistance in this case, and then there are these ampicillin resistant plasmid that have been added. Okay, and then since cells are given a heat shock at 42, some of the competent cells can take up this ampicillin resistant plasmid and get transformed. The treated cells are spread on agar plate containing ampicillin. Definitely, then only we can choose if what all cells have taken up the plasmid DNA, and then the cells are incubated for um from incubated for around 12 to 16 hours is also okay, or it can also be depending on what type of cells are there, what how many antibiotic selections. So not only in this case, in every case till now, I think we have looked at only one kind of antibiotic selection, like one kind of ampicillin, ampicillin, tetracycline, chloramphenicol. There can be dual antibiotic selection as well. So in that case, if there are two kinds of selection, like two antibiotics as selection pressure or antibiotic selective stress is given, then so the cells can take longer than 16 hours, longer than their usual time to really grow on that particular medium. Okay, having two antibiotics. So yeah, only colonies of those bacterial cells that have transformed or that have taken up the foreign DNA or plasmid DNA can will be able to grow in that nutrient medium containing that particular antibiotic. Now, when we talk about how to really calculate the transformation efficiency, So when here, see, the transformants will grow. There will be some number of colonies here present on the bacterial plate. So this gives us the number of transformants. Now divided by microgram of DNA that was introduced initially. Okay. Like here, when this one. So here we are introducing the foreign DNA or plasmid or recombinant vector, right? So how much of that recombinant vector did you really add? So there will be a certain amount, micrograms or nanograms, whatever will be the amount. So that will be there, that has been introduced into the solution. Okay. So that will come here. And final volumetric recovery. Recovery means when you have given heat shock and everything. Now, ideally, those small cell, like small volume of cells, competent cells have taken up your DNA. Now, those particular cells have to be given a proper space and nutrients and time for recovering from that shock, right? So, as well as dividing, replicating multiple number of times. So, what will happen is this recovery is in liquid broth, and then from that liquid broth, which is usually people do it in between 900, 800 microliters to 1 ml. So the whole 1 ml culture can't really be plated on your bacterial uh, on your bacterial plate or agar plate. So what happens is out of the 1 ml culture, people take suppose maybe 100 microliters, somewhere between 50 to 50 microliters to 100 microliters, and then plate that particular suspension, bacterial suspension, on the agar plate. Okay. So if we have to really calculate how many number of transformants have grown, then we should also be taking this particular volume in mind, that what was the final volume during recovery, or when we are trying to add, like culture these cells in liquid broth or LB medium here in this case, and then how much of that volume from that recovery volume was plated actually. Okay. So based on that, we can calculate the number of transformants. Now there are different factors that actually affect the transformation efficiency or uptake of the DNA by the cells. Okay. What happens here is, what is the amount of the plasmid that was used in the transformation in micrograms? This is what we are talking about. Okay. Now suppose there are good number of cells, recipient cells, but then the amount of DNA that you have added, that is low. So the number of cells that will take up that foreign DNA will definitely be lower. Okay. So what is the common amount of plasmid we use for transformation is 10 nanograms or 0.01 micrograms. 10 to 15 nanogram, it's good enough. So the final volume of the cell suspension at recovery. What this includes is, this includes the volume of the competent cells and the recovery broth. Here we are talking about the LB medium or the culture medium that we are talking about, liquid culture medium. And the volume of cells plated on the nutrient agar, since most of the time we will definitely not plate or it's not possible to plate the entire volume of the transformed cells. Suppose even if we plate the entire volume, so you won't really be getting distinct colonies. Okay. It will be a total smear and a lawn formation. So definitely then that won't be of any use to us. Okay. So a certain volume, this plated volume, will allow us to determine the proportion of cells that were plated and the number of colonies on the plate. Uh, this is represented by each colony, wherein if a colony has come up, then definitely that those cells have taken up that DNA. Those cells are transformed. Okay. Now, this is an example from the tutorial. I mean, from the lecture video only. If you have transformed 1 microliter of this much plasmid, so that means from here what you will get is how much amount of plasmid did you put in. So this will be 0.01 nanogram of plasmid you put, because this is the concentration of the plasmid, and like in 1 microliter, this much amount of DNA is present. Okay. Now if you are putting in 1 microliter only, then this much amount of DNA will be going in, and the number of volume of competent cells is 100 microliters. Okay. Now what happens is, you add this much amount of culture medium. This is the recovery medium that we were talking about, to your cells to get a total reaction of 1 ml. Okay. And then out of this 1 ml that I was talking about in the previous slide, you are plating 100 microliters of the transformed cells. Whether it's transformed or not, we don't really know. We will get to know only after seeing the agar plate. But still, like considering that uh, those cells after recovery, 100 microliters are plated, and we get 450 colonies. So in this scenario, what will be the transformation efficiency? It will be what is the microgram of DNA plated? This is the microgram of DNA plated giving us, and this is the final volume at recovery divided by the total volume, this one. Okay, and then efficiency, how much colonies did you get divided by microgram of DNA, and this is CFU refers to colony forming units. Okay. So this is the efficiency, transformation efficiency in this example. Now here we talked about chemical transformation. Okay, like we were using temperature changes and calcium treatment. Now there's electroporation as well, wherein you use electric fields to create force in the membrane so that the cell can pick up the DNA, foreign DNA. So what is happening? Suppose this is the DNA or some other molecule that you want to introduce, some other nanoparticle or other things, or a complex that you want to introduce inside the cell. This is the cell membrane. So material is introduced to the fluid around the cell, first of all, and then pulse is given to the whole uh, system, and that pulsed power generates an electric field that opens pores in the cell membrane, and the material that was in the vicinity will be taken up. Okay. And once the pulse ends, the pores can reseal or can actually go back to the natural state, and the material can remain inside the cell. This is electroporation. So in general laboratories, what happens is there are cuvettes which have electrodes attached, and there is a system wherein you directly pour the cell, like the in the previous one, you were actually mixing it one by one, the plasmid DNA competent cells. Here, in that particular, there's a cuvette wherein you put in your competent cells, you put in your DNA, and that particular cuvette has electrodes attached. You put in inside a pulse generator or system, and then the pulse is generated, and suddenly pores are created. Like in like very small amount of time, the DNA can be taken up by the cells via the pores created, which are actually transient, because once the pulse ends, the pores then reseal. Okay. So, what are the different factors that can affect transformation efficiency? Plasmid size that we were talking about, whether it's like really large or small. Definitely, if something which is around 1 kb, 500 bases long, that can easily get inside. Okay, definitely, but then plasmid size versus the vector size that we are talking about. Okay, that was there. Forms of DNA, whether it's supercoiled or whether it's relaxed, definitely the supercoiled one will have a smooth transition or smooth entry into the recipient cells as compared to the relaxed one. And growth of cells, whether the cells, like what is the, like whether the cells are really competent enough to take up the cells. If the competency is not good, then also your transformation efficiency will be bad or less. Okay. And like in the transformation protocol, what was the heat shock time given, what was the temperature, uh, how sterile or how like long was the recovery time given, whether the growth media was really sterile, chemical treatment given was proper or not, if there were any contaminants present in the chemical. So all these will affect the transformation efficiency, and definitely if there's any damage to the DNA, okay, then also uh, it won't be getting inside properly or taken up by the recipient cells properly. Okay. And so this we talked about normal chemical transformation, electroporation, and now we talk about chemical transfection. The principle is that you actually the one I was talking about, that there's a carrier molecule or you coat that whole DNA complex with polymers, certain polymers having a defined size of a definite size, and This coating, this polymer coating can actually facilitate the interaction of the precipitate. Precipitate means the complex that has been formed with the plasma membrane, and then this whole complex is taken up by the cell via endocytosis. Okay, so there are multiple chemical compounds that have been discovered which are able to make complex these complexes that we are talking about, and can deliver the DNA directly into mammalian cells. So this is actually incorporated in case of mammalian cells. In case of simple bacterial cells, transformation is fine. If transformation or electroporation is good enough. Now there's calcium phosphate treatment as well. There is like wherein there's DNA is mixed with calcium chloride in phosphate buffer and incubated for a certain time. And then this transfection mixture, the DNA coated with a polymer is introduced. Suppose in this case, DNA calcium phosphate complex is formed, it precipitates and it gets deposited on the cell surface. And then this particular complex, this deposited complex is taken up or endocytosed by the cells. Okay. Now, again, see here, the successful transfection can actually depend on the DNA phosphate complex particle size. Definitely, the size always matters here, which is very difficult to control because here randomly the precipitate or that complex between DNA and calcium phosphate is getting formed. So, how big that particular complex is, that can't really be controlled. It's totally random. Okay. Now, the disadvantage of this calcium phosphate method can be severe physical damage because this complex is getting precipitated on the cell surface. Okay. Damage to the cellular integrity due to particulate matter settling on the cell, and which also reduces the cellular viability and can add to the cytotoxicity. Okay. And eventually kill the cell. This can also be happened. This treatment can also has proven to be toxic in some cases to the cells. Then there's polyplexes method, where the DNA is complexed with a chemical agent to form soluble precipitate, wherein this particular chemical agent actually electrostatically, like this coulombic interactions with the DNA and some other polycationic carbohydrate, DEAE. So DEA is actually uh, basically DEA is a matrix or a polymer having slightly positive, like it is also used during exchanging or purifying some anionic proteins or basically anionic nucleic acids, mainly RNA or DNA or tRNA, mRNA, like that. Okay, positively charged cationic lipids. But in this case, the EAE can be, because this is cationic in nature, having positive charge, this can interact with the DNA. Some positively charged cationic lipids, transfectin is one example. There can be polyamines, which are again positively charged. These are used for this purpose. Okay. So what happens is they form aggregates of DNA because they're interacting with the DNA, forming a polycationic complex that can be easily taken up by the cell. Okay. This is the same thing that has been shown here schematically. And there can be liposome or lipoplex complex again. Okay. So what happens here is DNA is transfected inside the or introduced in the animal cell, wherein DNA is packed in lipid vesicle or liposome. So if something is packed in a lipid vesicle, then through the lipid bilayer, that interaction between the lipid residues along with this particular lipid vesicle will be like really compatible and good enough to pass through. Definitely. Okay. So in this approach, the DNA containing vesicle will be fused with the cell membrane and the DNA will be delivered to the target cell. Okay. So this will be like, this will have like really less compatible issues because the DNA is getting packed inside the lipid vesicle or liposome. Okay. But then liposome has to be prepared like very carefully, because preparation of liposome and putting the DNA inside is a very crucial step to achieve good transfection efficiency. So liposome can be prepared with cationic or neutral lipid, which facilitates DNA binding to the complex. And definitely that the liposome that we are talking about should, because it has to hold the DNA inside. So it should be the characteristic of that particular liposome, or the lipids that are used to prepare that liposome should have characteristics or properties that help it to really bind to or keep the, like interact with the DNA stably, right? And so that and finally the whole that complex, the liposome complex, is taken up very easily by the cells. So the lipoplex method is applicable to a really wide variety of cells and found to transfect large size DNA as well. So another advantage is because with the addition of ligand in the lipid bilayer, it can be used to target specific organ in the animal or a site within an organ. Okay. And then there's transduction, wherein you actually use viruses to introduce your genetic material, any foreign genetic material. So what happens here is, see, Uh, these phages, bacteriophages, what happens here is bacterial chromosomes are there in the host bacterial cell, and these viruses infect the bacterial cell. So what happens is phage enzymes break down the host DNA, and the cell can create new phages, including phage and the host DNA. And then what happens here is, if this is the general infection mechanism. Okay. Now this phages, bacteriophage viruses have, if something of if a DNA of our interest is introduced into these viruses or bacteriophages, so this virus can then be like naturally made to affect like infect the bacterial cell, and then when the bacteria it gets inside the bacterial cell, then it can introduce that particular foreign genetic material easily. Okay, so transducing the phage insert donor DNA, it can actually put inside the donor DNA, and this DNA can be included in the recipient chromosome when it can actually recombine with the recipient chromosome and get introduced inside the bacterial cell or recipient cell. Okay, this is what has been shown here. Now, so we talked about cloning. First of all, we made the recombinant vector. Then we talked about how to deliver that recombinant vector via like different means. And then if that recombinant vector has actually gotten inside the cells, how do you really screen it? Okay, because some cells will be taking up that foreign genetic material, some won't be taking up the foreign genetic material, or even if some cells have taken up the foreign genetic material, it didn't really get inserted with the genome. So that won't make any sense in various types of experiments. Okay. So how do you really screen the recombinant clones? Okay. So there can be enzymatic screens, there can be antibiotic resistance gene. I think we have talked about antibiotic resistance gene a lot. There can be some phenotypic changes. If you guys remember from the last tutorial session, we were talking about galactosidase enzyme or LAC reporter gene, or blue-white screening of bacterial cells. Okay, so enzyme, antibiotic resistance gene, or phenotypic changes. Okay, now antibiotic sensitivity. What will happen is, suppose this is the ampicillin resistant. This is the portion of the gene which codes for ampicillin resistance. Okay, which renders the uh, genetic, or if it is inside the cell, inside the organism, it renders the organism resistant to ampicillin. Okay. Now this is the insert, and this is the vector having ampicillin resistant gene. Okay. Now, if this is getting transformed, then only the colonies, those colonies will grow, or those cells will grow, which have ampicillin resistance, which means this has gotten inside. Okay. If this particular insert is not at all there, and only like this particular, like if this is getting transformed, then this won't be having any colonies. Okay. So what is happening here is, if this particular insert is there, and this recombinant clone gets inside, then ampicillin resistant colonies will grow, otherwise it won't. So also there are so we were talking about I think last class also we were talking about different kinds of antibiotics that are there and what is the product like what is the like how do bacterial cells really uh, what is the product that those resistant genes code for so that these become like like they lose the cells bacterial cells lose their sensitivity towards these antibiotics. Okay, so there's ampicillin which codes for ampicillin resistant gene codes for beta lactamase which degrades ampicillin. Okay, sometimes what happens is beta lactamase some when you keep on growing or not only keep on growing, suppose some if for a long time you have grown cells on a plate containing ampicillin, what happens is after a certain point of time you will see like there are bacteria circular bacterial colonies, and outside the bacterial colonies you will see a ring like thing outside. So what happens is those are actually sometimes after a long period of time those cells start producing because they suppose if some of the mutant cells are there which actually uh, somehow have ampicillin resistant gene, then that particular gene codes for beta lactamase enzyme. Okay, and that lactamase, beta lactamase degrades the ampicillin. So what happens is the whole nutrient medium or the whole agar plate has ampicillin, and the colonies which have grown there will eventually start releasing uh, like beta lactamase enzyme, and that enzyme acts on the ampicillin in the medium and starts creating a ring-like structure around the colony, which shows that like the beta like degradation of ampicillin has taken place. Then there's kanamycin, and a kanamycin resistant gene codes for neomycin phosphotransferase 2, which actually modifies the kanamycin and renders it inactive. So what happens is kanamycin actually binds to the ribosomes and stops the translation, interferes with the translation of proteins. So what happens is if this that particular resistant gene codes for this particular enzyme, and this enzyme introduces a modification which is covalent in nature on the kanamycin. So kanamycin can't really go in and bind at its site on the ribosome. Okay, so then the particular cells actually lose their sensitivity to this particular antibiotic and become resistant. Yes, tetracycline. So the tetracycline resistant genes produce ribosomal protection proteins. I think this question was there from the previous lecture videos, like previous assignment. And what happens is these are certain proteins which help in removing tetracycline outside the bacterial cell. Now also there's chloramphenicol resistant genes, codes for chloramphenicol acetyl transferase, and chloramphenicol gets changed or modified to acetyl, gets acetylated acetyl chloramphenicol. Okay. Insertion and inactivation we had talked about in the previous class. Okay. So in this approach, what happens is a foreign DNA is cloned within the coding gene, which is responsible for a particular phenotype. Suppose we have been talking about antibiotic resistance. So if something is kanamycin resistant, okay. So the phenotype in this example is kanamycin resistance. Now, as a result of the insertion, if something gets inserted within the kanamycin resistant gene, then the particular cell will lose its resistance to kanamycin and will become sensitive to the antibiotic. So if we try to grow that particular cell in a kanamycin containing medium, then those cells won't be able to grow. So in this approach, a foreign DNA is cloned within the coding gene responsible for a phenotype. As a result of insertion, the gene product is not available to modulate the phenotype of the host. Now, this approach is called insertional inactivation and can be used with a suitable genetic system. Now, I think in the plasmid vector we were talking about in the last tutorial session, PBR322, it has two uh, like resistance genes. One is ampicillin resistant gene and tetracycline resistant gene. So suppose that this PBR322, if a gene fragment will be cloned in SCA1, okay, and it will disrupt the ampicillin resistant gene. Okay. So what will happen is the clone will now become sensitive to ampicillin, but it will still retain resistive resistant resistance to tetracycline. Okay, but the original plasmid will be resistant to both these antibiotics. Now, in order to select the clone, first the transformed E. coli is plated on the tetracycline containing medium, and then a replica plate will be made on ampicillin containing medium to identify the clone growing on tetracycline media but not on ampicillin media. This is also from the previous class. This is what was being talked about. So something which has uh, which cannot grow on ampicillin or tetracycline medium, that means it has not taken up the DNA at all. It is not at all transformed. But transformed ones will have only transformed colonies which can grow in either ampicillin or tetracycline containing medium, because both of the whether it has whether the gene has inserted at its correct position or not, the whole plasmid has been taken up by the cells, and that means it can grow in either of the medium. Okay. Now, if transformation has occurred with the genetic construct at its like not exactly at its position, or what we can say is transformation in with non-recombinants, then what will happen is it will grow in both ampicillin and tetracycline containing medium because both the resistance genes are intact. But if the foreign DNA, foreign genetic sequence has inserted at its correct position where we actually intended to initially, then what will happen is the recombinant vector transformation with the recombinant vector will make the cell sensitive to one ampicillin. One antibiotic. Suppose ampicillin resistance gene, if the insert has gone there inside the ampicillin resistance gene, then what will happen is that particular cell will become sensitive to ampicillin. Okay. So here in this case, now here the case is the gene insert is getting inserted at the in between tetracycline resistance gene. So if it is getting actually inserted correctly at its location, then the tetracycline resistance will be lost definitely, and the cells will become sensitive to tetracycline. So here in this case, we are able, like using insertional inactivation, we are actually able to separate transformants with transformants from non-transformants in general, but with using insertional inactivation, you can actually differentiate between recombinants and non-recombinants as well. Okay. So this is the alpha complementation process we are talking about. So lactose in the surrounding medium triggers the laxi-operon in E. coli, okay, which leads to that ZYA genes are there. So beta galactosidase is coded or expressed, and production of beta galactosidase metabolizes the lactose. And I like in the natural case scenario, beta galactosidase is a tetramer, homotetramer containing like all the four subunits are the same. And in case of suppose hemoglobin, this is heterotetramer because there are two alpha and two beta subunits, right? Now in its active state, beta galactosidase is tetrameric in nature. Now what happens is there's a certain deletion, uh, like LAC-Z delta M15, which codes for omega peptide, wherein the first N-terminal residues are deleted. Okay, like the gene which codes for the first few N-terminal residues that has been those particular that particular region has been removed. So this omega peptide, which has actually deleted N-terminal residues, are unable to form this tetramer of beta galactosidase, and therefore it is inactive. Okay. Now, in order to like really regain its active tetrameric state in the presence of N-terminal fragment of the protein, the alpha peptide is introduced. This particular N-terminal fragment of the protein is introduced. Okay. Now, individually, if we talk about that only these many N-terminal residues, the alpha peptide or the omega peptide with the deleted N-terminal residues, individually, neither of them are functional by itself. Okay. Now, only when these two are brought together, then only it can really get into its active state, forming the homotetramer and code for a functional as in and function as a like perform the function of a functional beta galactosidase. A plasmid containing LAGZ alpha sequence, if we are actually transforming or introducing a plasmid coding for this alpha peptide and inside the cells which actually code for omega peptide, which can't really which have this particular deletion for the N-terminal part of the protein, then the two peptides are expressed together in the cells and thereby forming functional beta galactosidase. So the rescue of the function of the mutant beta galactosidase, mutant in this case is the omega peptide, by the alpha peptide is known as alpha complementation. Okay. Now, how is this used for screening the recombinant colonies? Okay. What happens is, suppose a multiple cloning site is present within the LAGZ sequence in the plasmid vector. Now, this sequence can be uh, digested by restriction enzymes to insert your DNA of interest or any foreign DNA. Now, when a plasmid vector containing this particular, like containing your gene of interest or foreign DNA is taken up by the host or the recipient cells, then alpha complementation won't occur. Therefore, a functional beta galactosidase enzyme won't be produced. But if the foreign DNA is not inserted into the vector or if it is inserted at a location other than the multiple cloning site, then what will happen is the LAGZ gene in the plasmid vector which codes for the alpha peptide, will complement the LAGZ deletion mutation, which codes for the omega peptide, and then alpha peptide and omega peptide together will produce a functional beta galactosidase. Okay. Now, if functional beta galactosidase is being produced, that is being used to act upon a substrate called XGAL, which is a which gives us a which is actually a chromogenic substrate, which generates a blue colored substrate. Okay, if beta galactosidase is functional. If it is not functional, then that blue colored substrate is not produced. And this is what is used during screening of recombinants. Okay. So this is actually called blue-white screening because you are screening the clones with recombinant DNA using XCAL substrate, chromogenic substrate, which is added to the nutrient medium or to the agar plate. So beta galactosidase produced, XCAL hydrolyzed to this particular substance which dimerizes to form an insoluble blue pigment. Now, colonies which are formed by non-recombinant cells will definitely will code for both, um, will have both the alpha peptide as well as the omega peptide. So, these will appear blue in color, while the recombinant ones will appear white. Now, desired recombinant colonies can be easily picked and cultured, and in order to induce the expression IPTG, which is an analog of galactose, it induces the expression of LAGZ gene. This is the thing that should be kept in mind is this is not a substrate for beta galactosidase, but it acts only as an inducer. Okay, and but for visual screening purposes, you have to use a chromogenic substrate like XCAL. So this is how the plate looks like. So these blue ones have to be neglected because these blue colonies, the cells in these colonies haven't really taken up your DNA. Okay, or even if they have taken up the DNA, the insert hasn't been placed exactly at the correct site. Okay, so this is what is represented here. This is the plasmid vector. This is the foreign DNA. If the foreign DNA is inserted within the LAGZ, then functional beta galactosidase is not there. White colonies will be produced. If it is inserted, it has been taken up and it has been inserted, but then it has not been inserted at the correct site or outside the LAGZ position, then there will be blue colonies, or it hasn't been taken up at all, then also it will give us blue colonies. Okay. So only the recombinant ones can be chosen from all these colonies. Now, here, one thing to be taken care of is complementation of mutation. So, two types of selection plays a role here. One is positive selection, wherein the host strain, wherein we are trying to, or the recipient cell. Okay, the host strain can't grow on the media lacking the functional gene. But if the host cell or the recipient cell is transformed with a recombinant clone, that is able to supply the gene product which is required for its growth, required for the growth of the recipient cells here. And in negative selection, what happens is a chemical compound is added to the medium, which will be converted to the suppose a certain gene that has been introduced that codes for an enzyme or some other molecule. Now, what happens in this negative selection is if a chemical compound that is added to the media, only if those cells that have taken up the gene or the foreign gene, if those cells will be that foreign gene will be having certain sequence that codes for a cytotoxic agent. And only when the cells have taken up the like that gene product, then those cells, as a result, those cells won't be growing in this case. Similarly, if we are talking about blue-white screening also. So if we see white colonies are formed when non-functional beta galactosidase is there. So it will be like an example of negative selection also. Okay. Yeah, but host strain transformed with a recombinant clone has non-functional gene product. So what we saw in case of beta galactosidase, and it grows in the presence of the compound in the media or like non-functional gene product. So white colonies are formed. Okay, that can also be an example of negative selection because you are rendering that particular gene product non-functional and then selecting out your like recombinants and transformers. Now there's reporter gene assay. What happens is sometimes in a natural case scenario in the native cell, this is your promoter, this is your natural gene product, this will code for mRNA, and there will be the gene product, protein or uh, these things will be there which are endogenous in nature, like required for the native cell. But in case of a recombinant cell, what happens is a strong promoter is like under the same promoter, the reporter gene is uh, fused, and then this will now if this promoter is a stronger one, the same promoter is a stronger one, this will code for like drive the expression, like drive the expression and make the genetic sequence to code in large expect like lead to the much larger expression of this particular genetic sequence, and this transcripts will be again coding for or expressing the reporter molecules. Okay. In this case, it's reporter protein, enzyme, GFP, etc. Okay. Now, this is the reporter gene assay. So, if this has been fused with your natural gene construct, now what will happen here is, uh, if this reporter gene is getting transcribed or translated, then your whole gene is getting expressed. So this is the luciferase reporter assay. What happens here is the gene, like there is luciferase enzyme, the gene encoding luciferase is, suppose this is the reporter gene, okay, that has been fused here. And then what happens here is there is an external substrate luciferin which is added, and in the presence of ATP and magnesium ions, what happens is luciferin gets converted into luciferyl adenylate, and AMP is incorporated. And when if luciferase is being encoded from the reporter gene assay, reporter gene construct, then in the presence of a suitable wavelength of light as well as in the presence of oxygen, this particular luciferyl adenylate gets oxidized to oxyluciferin. Okay, and then uh, this decarboxylation reaction, this particular light generated is actually uh, of our interest, wherein we get the signal. Like this gives us an idea that okay, our gene of interest is getting expressed, or like it has been incorporated into the cells. This is what is happening here. Luciferase expressing vector is transfected into the cells of our interest. Suppose this is the eukaryotic cell. This is the plasmid vector. The nucleus is there, And what you do is you actually lyse the cells and then dispense it into uh, like it can be cuvette, it can be 96 well plate, and then you add luciferin in this. This is the substrate which will be acted upon by luciferase enzyme. Now, in this case, see here it's written positive control and negative control. Positive control is something that will definitively give the result, that will definitely give the signal required, showing the presence of like a known gene construct having luciferase encoding sequence. And negative control will be anything that won't be giving us, that we know for sure that won't be giving us any uh, or won't be giving us any signal due to luciferase activity. It can either be your buffer or um, your simple cell suspension, any cell without having any luciferase activity, any kind of negative control is possible. Okay, and then after adding luciferin substrate into your samples, then the luciferase enzyme, if the cells have actually taken up that particular vector, then the luciferase enzyme being encoded will convert the luciferin, will oxidize and decarboxylate this particular chemical, thereby giving off light or signal. And this particular signal will be detected using this instrument, and we will get a luminescent curve or luminescence graph. Okay. So this is luciferase reporter acid. And now after, okay, you have screened your things also. Now there's also another way to confirm that whether your foreign or gene, the foreign DNA or DNA of interest has actually gotten inside your recipient cells. There's Sanger sequencing or Sanger sequencing method or di-deoxy chain termination method. What happens here is this method is originally developed by Frederick Sanger, and a single stranded DNA is used as a template to synthesize a complementary copy with the help of a DNA polymerase and in the presence of nucleotides. Now, what happens here is, in general, we use DNTPs or deoxynucleotides. Okay. Deoxynucleotides, we use it here in case of like only one part of the 2 prime uh, is deoxy, like there's no oxygen present. But what happens here is, in case of Sanger sequencing, we use uh, nucleotides which are uh, missing, like which do not have oxygen at 2 positions, at 2 prime and 3 prime positions. Okay. So what will happen is, with the 3 prime hydroxyl group, the like chain extension takes place. Now, if that OH group is not there, then the chain elongation won't be possible, or chain polymerization won't be possible. So what happens is, if both the like 2 prime was already is already without oxygen in case of DNA, but if the 3 prime is also not having the hydroxyl group or OH group, what will happen is there won't be further extension. The chain extension will be stopped at that point where where the that particular nucleotide has been incorporated with a missing 2 prime OH as well as 3 prime OH. Okay. So this is what is written here. Like, the polymerization reaction contains a primer and nucleotides, 3 normal nucleotides, 3 normal, like out of the 4 nucleotides, each reaction tube will be having one set, like one type of uh, nucleotide having both, like where both the 2 prime as well as the 3 prime OH groups are absent. And so that is called DDN. Okay, DDNTP or di-deoxynucleotide triphosphates. Now, when DNA polymerase utilizes DDNTPs as nucleotide, it gets incorporated into the clone chain, but chain elongation stops at DDNTPs due to the absence of 3' hydroxyl group. And if 4 different DDNTPs are taken into account and taken into 4 separate reactions, then after the reaction is finished, each of the gel content is ran on whether it's agarose or polyacrylamide gels, and then we look at the length of the DNA being formed, okay, or the fragments taken into consideration. Okay, so how do you really analyze a Sanger sequencing results? So this is suppose your original DNA construct. A reaction means DD ATP has been introduced. B reaction means TTP has been introduced. DD-GTP has been introduced in G reaction, and in C reaction too, DD-CTP has been introduced. Now, wherever like there's this strand. Now, if DD-ATP is there, then it will stop at this part only. Like only A and T will be formed. Then it will stop at this part. Okay. So similarly, for the other reaction tubes, this is how different fragments will be generated. And then starting from the lowest fragment, which is running at the lowermost point of the gel, that will, the smallest fragment will run the fastest. And accordingly, we can get an idea from each tube. Like if this is the A tube, then it will have like A and all these things here. And then, and when you really compare all the four lanes having the different fragments, then you can come up with what point the reaction got stopped, and you can actually eventually one by one get the whole sequence of the DNA that was being sequenced. Now there's Maxim Gilbert method also, which is here you actually modify the bases, each kinds of bases. Like there we were introducing like all the four bases having missing 3' hydroxyl groups. In this case, like there are like certain chemical modifications being introduced into the bases, and then the modified bases are actually uh, taken care of, or using specific chemicals. Those are actually cleaved off. Okay. So in this method, 3' or 5' radio labeled DNA is treated with base specific chemicals, which randomly cleaves the DNA at their specific target molecule, generating fragments. Okay. Now these fragments are analyzed on gels, polyacrylamide gels, and autoradiogram is developed, and the fragment with terminal radio label appears as a band in the gel. So this is what happens. The fragment in the G lane. Now, this is G reaction, G plus A reaction, T plus C reaction, and C reaction. Wherein, like in G reaction, only the G residues are modified. In G plus A reaction, both these residues are modified. T plus C reaction, T as well as C residues are modified, and C reaction, only the C residue is modified. Now, when all these reaction tubes are run on acrylamide gels or acrose gels, what will happen is G and G plus A. So when we compare both these links, then the one like G plus A band, when it is having certain extra band, then the fragment in G lane is read as G, whereas fragment present in G plus A but absent in G will be read as A, definitely. Similarly, if T plus C and C, the fragment that is present in both the lanes will be read as C, but something extra that is present in T plus C will be read as T residue. Okay. So the fragment present in T plus C but absent in C. In this case, suppose this one. Okay. See, this is being read as T here. Okay. To get the DNA sequence, the band with the lowest molecular weight is read first. See, both of them have the band present here. So this is G. Okay. Now next, this is present here, but the band is not present here. Then this will be A. So G followed by A. Then comes the next molecular weight band that is coming up is this one, which is extra as compared to the C lane. So this will be uh, like this is C, but then this will be as in this is the C, like both the C residues are getting modified here, but here T residues are also getting modified. Okay, and cleaved off from that point. So this will be T. But here both C residues are present here. This will be GATC. Okay. Similarly, you can come up with the whole like nucleotide composition of your DNA sequence. Okay. Now let us move on to the few questions, example questions. Okay. So people are here. Now, so which of the following agents is used to make E. coli competent cells? Can anyone try and answer? Calcium chloride. Yes, good. Calcium chloride. Okay. Next, while making competent cells, it is essential that the bacteria is in yes, log phase or exponential phase. Definitely because we need like actively when actively growing cells having all the factors present inside so that when the DNA is taken up, they can actually those are arrested at that particular phase so they can actually replicate later when the DNA is taken up inside. Okay. So this is option B, log phase or exponential phase. Transformation efficiency in E. coli is highest for which type of DNA? Like the more compact the DNA is, the transformation efficiency will be higher. Okay. So out of these four options, which is having the more compact form? Yes. Yeah. So option A will be the answer. Okay. Because this is the more compact for in this case. Okay. Because the linear DNA or open circular DNA will be the most problematic ones. Okay. Now, which of the following components block the non-specific sites on the cell wall and increase efficiency of plasmid uptake in yeast cells? Okay, so for this one, I was talking about like sometimes only if you're adding DNA that can be taken up by the competent cells. Now, sometimes that is not so easy in case of higher order organism in cells of higher organisms. So what happens is that DNA has to be packaged with some other cells or it has some other chemical treatments have to be given so that only the DNA of our interest is taken up. So in case of yeast cells, what happens is these are incubated with a mixture of lithium acetate salt is there, PEG, this PEG molecule is there. This value can range from 2000 to 4000 to 5000, 6000, and like 20,000 also. This is basically a crowding molecule. Okay. So it brings in different components together and helps in making a tighter complex. Okay. And then in case of yeast cells, single-stranded carrier DNA and foreign plasmid are all of these things are used for transformation and then heat shock is given for a much longer time. Okay. Now the purpose of adding carrier DNA is to block the non-specific sites on cell wall and make the plasmid DNA available for uptake. Okay. Post-transformation cells are pelleted to remove transformation mixture and resuspended in 1 ml water. And so the answer of this question will be what actually blocks the non-specific sites on the cell wall? Option will be option B, single-stranded DNA. Just give me a moment. I will bring my charger. Just a moment. So now, which of the following enzymes could be used to partially digest the cell wall of yeast cells? Can anyone try? It will be zymolase. Okay. Option C. Lysozymes for bacterial cells, digesting the bacterial cells. Okay. And in the calcium phosphate method, the mammalian cells uptake the DNA calcium phosphate complexes by which process? We discussed in the yes, yes. Endocytosis option. Sorry, this will be endocytosis. Okay. And what's pinocytosis? Can anyone answer? Pinocytosis is basically cell drinking. Okay. Uptake of molecules or small molecules or liquid medium from the extracellular matrix. Okay. Extracellular environment. Now, the method in which viruses are used for transformation in mammalian cells is yes. Option C, transduction. Now, in the back-to-fiction method, which type of organism is commonly used to transform plant cells? Can anyone try? Yes, yes. So, and what about Shigella and Salmonella? So, like in plant species, what happens is Agrobacterium tumefaciens is the most common uh, like back-to-fiction method being used, and in animal cells, uh, phagocytosis is done, and the bac- like host bacteria is actively taken up by the host cell, like the bacteria containing our gene of interest is taken up by the host cell either through phagocytosis, and then those are trapped inside phagosome, okay, which are membranous vesicles. And the bacteria which has gotten inside can escape from the phagosome and get lysed, and the DNA of our interest, which was incorporated inside the bacteria, can get released in the cytosol. Okay. So, see, the bacterial species used in this method in case of animal cells are usually Salmonella, Shigella, etc. And in case of plant cells, the same thing is Agrobacterium tumefaciens. Okay. So, correct option is option B. Now, when we were talking about insertional inactivation, in case of blue-white screening, the successful cloning of the gene of interest in the LAGZ gene will result in which type of colonies? Yes. And also, which type of selection was it? We were talking about positive selection and negative selection, right? Like something here, if the colonies are colorless, then that means the enzyme beta galactosidase in this context has been rendered non-functional, right? So this is a kind of negative selection. Okay. If somehow we were choosing for those recombinant colonies which are coding for the functional beta galactosidase, then in that scenario, and then they are giving us blue colonies, then in that scenario, it would have been a positive selection. Okay. Now, the cytotoxic compound which is added to the selection media for URA3 mediated negative selection. This is we are talking about in yeast. Okay. So for this question, what happens is we talked about positive and negative selection, right? So in this negative selection, if this gene has been introduced inside the yeast cells, what happens is this gene codes for this OMP decarboxylase, and this active enzyme, this particular 5-fluoroorotic acid is given inside in the nutrient medium, okay, in the growth medium. Now, if this enzyme coding gene has been incorporated inside the yeast cells, what happens is this will convert this particular substrate into a toxic substrate. Okay. So if those cells containing this particular genetic sequence or coding for URA3, what will happen is functional URA3 gene product will produce this toxin, and those cells containing the URA3 gene product won't be able to grow in a medium containing this particular substrate, 5-fluoroorotic acid. Okay. So this is again a negative selection. So the answer will be 5-fluoroorotic acid. Okay. Option A. Now, we just talked about Sanger sequencing. Dideoxynucleotides are used in which kind of sequencing? I mean, I told the answer already. So it's option C, Sanger sequencing. Okay. So in the G reaction of Sanger sequencing, the nucleotides that are used are what kind of nucleotides? Which set of nucleotides? Can anyone try? So in G reaction, we are option B. Yes. So in G reaction, we are actually stopping the reaction after the addition of G residue, which is which has both like where the 2' as well as the 3' hydroxyl groups are absent. Okay. So option B. Now, which of the following reaction is catalyzed by the gene product of 4A gene? Okay, so in this case, like this is the one we were talking about. So we looked at luciferase reporter assay, right? Then there's LAGZ reporter gene also, which codes for beta galactosidase. Now there's this alkaline phosphatase assay as well. This 4 gene codes for the actual gene product is alkaline phosphatase, wherein what reaction does it catalyze? It releases inorganic phosphate. Okay. So in this case, the answer will be option C, release of inorganic phosphate. Okay. Now, before the second, like last two questions, what happens here is there's another type of insertional inactivation. We were talking about blue-white screening, right? Now here there is this FAD screening as well. During an infection cycle, what happens is viruses can undergo either lytic or lysogenic stages. Okay. Now in the lytic phase, what happens is the host cell is lysed completely to release the virus particle or viral particles, whereas in the lysogenic stage, the virus can replicate without the host cell getting lysed. Okay. Now there is the CI repressor gene which codes for CI repressor, and this particular CI repressor is responsible for forming lysogens. Okay. Now, if this CI gene is functional, what will happen is the plaque that are formed will contain analyzed host cells and will give a turbid appearance. Whereas, if the if the gene, CI gene or CI repressor coding gene is somehow inactivated due to insertion of, suppose, a foreign gene or foreign DNA of our interest, then what will happen is the this turbidity won't be there. Okay. So this feature can be used to screen the clones to detect a functional CI gene or absence of a clone or absence of CI gene, like absence of a functional CI repressor, which means the insert has been inserted at that CI repressor encoding gene. Okay. Now, so this is the question that follows, that the CI repressor when active, what is it? We just looked at the option, like the concept. It actually represses the lytic phase. Okay. Option B. Now, this is an exam, like question regarding transformation efficiency. So, this is the question wherein this much amount of DNA was added to transform this much volume of competent cells. Okay. Now, the cells were recovered using this much LB media so that the final volume became 1 ml. Okay. And out of this 1 ml recovery volume, 50 microliter was plated, and number of transformed colonies we got was 500. Okay. So can anyone calculate and let me know the transformation efficiency? Like you guys can try out, then I will be there to tell the answer. So has anyone solved? So what is given here is how many transformed colonies we got? 500. Okay. Now 500, and how much of DNA was introduced? This much microgram, which means 10 to the power minus 3 microgram. So 500 divided by 10 to the power minus 3. Okay. Then what was the final volume? It was 1 ml. Okay. 1 ml, or you can also write as 1000 microliters. So 500 divided by 10 to the power minus 3 into 1000 divided by how much was plated out of the recovery volume? 50 microliters, right? So this will give us 1000 by 50 into 500 number of transformants divided by the amount of DNA that was introduced, right? So then this will give us 10 to the power 7 colony forming units per microgram of DNA. Okay. Answer will be option B. Okay. So this was it for today's class. Next week, we will be summarizing the week 6 videos, and then solving few assignments. Okay. Thank you for joining. See you the next week. Okay. Thank you, everyone. Yeah.